I'm conscious I'm standing between you and coffee, so I won't keep you too long. I'm going to tell you one story from our group that has recently been published, where we're looking at sort of, I guess, different ways to treat bacterial infections. And I'm very lucky that a close colleague of mine, Kevin Kavanagh, who spoke actually in this track earlier, has worked with us on this problem. Because really, we're a chemistry group. So fundamentally, we are a synthetic chemistry group where we make small molecules most often, but we also work a little bit with metals, nanomaterials, and soft materials. But what we like to do, essentially, is to not just synthesize them, but then make things that if we tickle them with something, that we can make them do a trick. So, for example, the one I'm going to talk to you today is where we have a small organic molecule that we give a chemical input, and only with the aid of that chemical input do you get a biological response, in this case an antimicrobial response. But very simply, it's a chemical or physical input to these molecules that gives you something that you can measure. And so I trained in a very fundamental field of chemistry, what's known as supramolecular chemistry, and anyone who is familiar with that type of work, this is a very classic design where you have some sort of a synthetic receptor which is capable of binding to an analyte or a molecule. In this case, I'm talking about an ion, in particular an anion, so something that's negatively charged, and you form this ion receptor complex. And this has been sort of knocking around as a very fundamental type of chemistry for a long time, but over the last few years, I suppose, it's moved a little bit more towards, well, once you bind that ion, can you also, utilize that ion receptor complex to do something more interesting? And one of the fields that has kind of mushroomed out from that is the idea of anion transport. So where you're capable of not just binding that ion, but also facilitating the transport of it through a membrane. And in general, membranes are impermeable to ions. And if you want to have ion flux, you rely on either protein channels or carrier proteins. And so there's a number of examples of disease states that are characterized by misregulation of ion transport. The one that springs to mind most often is cystic fibrosis, which results from a malfunctioning chloride transporter protein, which gives rise to the symptoms of that disease. So very often misregulation in ion flux gives rise to types of disease. But this is very simply the sort of concept where you have some sort of a donut-type receptor that binds to the ion and then is able to facilitate the ion through the membrane in this mobile ion carrier type of a schematic. And so a lot of the work that I'm going to talk to you today was sort of inspired by work of a collaborator of ours, Phil Gale, who's in the UT Sydney, where he demonstrated that with a very simple molecule that is capable of binding and efficiently transporting chloride through a membrane, was able to raise the pH of a lysosome by disrupting the chloride homeostasis. And so essentially increasing the concentration of sodium chloride inside the cell results in a whole load of downstream biological effects, but ultimately potent cytotoxicity. So it's I guess, a different way of looking at, this was sort of touted as a potential strategy for anti-cancer research, but again, obviously you have the problem of non-specific disruption of chloride homeostasis is not ideal. And so we took this idea and we work very often with this unusual, so for synthetic or for chemists who like to make things, they'll know that four-membered rings are very difficult to make and very often are very unstable. And this compound that we know as squaramides are four-membered cyclobutene rings, but they're aromatic. And so this aromaticity makes them very, very stable. But not alone that, they have really useful properties, and they have been used in the literature. We reviewed this a few years ago now, but things as diverse as organocatalysis, self-assembly networks used in medicinal chemistry, bioconjugation, and molecular sensing. And it most often comes down to these, so these are two nitrogen atoms, two NHs, that are very strong hydrogen bond donors, so they're sticky, and particularly sticky to things that are negatively charged, so anions. And so they are, in many ways, an ideal anion recognition motif. They're able to bind very effectively to negatively charged species. And this is a urea, and this is a thiourea. And again, in the literature, these have been used extensively for ion binding. So very simply forming these ion receptor complexes. But squaramides in more recent times have been shown to be much more efficacious. They have a lower pKa. So these NHs are much more acidic, which means they're stronger. They've got a stronger hydrogen bond donating ability. And also the fact that, and this has been shown in several studies now, that the cyclobutene ring is aromatic, but when it binds to something, the aromaticity of that system increases. So there's a second thermodynamic driving force towards recognition. And so while we were, I guess, investigating a lot of the fundamental aspects of anion recognition, anion binding, it was Luke Brennan who has done all of the work that or the vast majority of the work that I'm going to talk to you about, is that a lot of the known antimicrobials on the market are actually cation transporters. So these are metal transporters. So monensin, which is used heavily in the US for feedstocks, so as an antimicrobial or an antibiotic in feedstocks, is a sodium transporter. Valinomycin, who is clinically used antimicrobial, is a potassium transporter. But up until now, there's been almost no examples of anion transporters and their potential as antimicrobials. So it kind of stands to reason that if you can disrupt metal homeostasis, if you can also disrupt anion homeostasis, you might end up with a similar effect. And antimicrobial resistance, again, it's everywhere. You can't miss it. It's sort of on the top priority of many of the lists in terms of new therapeutics that are required on the WHO's list of priorities for research. But there was a scarcity. There was about 3 that I could find at the time, and these were just compounds that just so happened to be ion transporters and also showed the fact that they could have some level of antimicrobial ability. But the gap in between and trying to understand, well, where is that coming from, was sort of lacking. So we set out to make these relatively simple molecules as a proof of principle that we could make these efficient anion binders that would also transport, with the idea that potentially they could also be antimicrobials. So this was our design, where you have now an NH, but we've replaced it with a CH to enable to incorporate a fluorescent tag. So these compounds are fluorescent, so we can track their uptake and localization on that basis. And we can also really mess around with the functionalization of these. So it's not necessarily looking at a drug design element of this, but just being able to play around with the functionality will help us to increase either ion binding or make them more lipophilic, which very often makes them better ion transporters. And so we started out with these 4 compounds, and the idea was that we were going to compare them against this compound 5 and 6, which were the very cytotoxic compounds that had been previously reported by Phil Gale. And so I won't labor over the synthesis of these. It's very simple. 2 steps. Goes from a commercially available starting material in moderate yields to the range of target structures 4 initially. We could see straight away from just looking at these under UV that they're very brightly green fluorescent. And also for the structural people out there, there's potential isomerism around this CC bond. And in order for this to be an effective binding cavity, the trans configuration of this double bond is required. And what we've seen from crystal structure analysis is that it is, in all cases, it's the trans configuration. And again, so this is a crystal structure, and it's very clear to see. This is a nice pocket. So this is a DMSO molecule. It's not an anion, but it is a very good hydrogen bond acceptor. And so if you've got 2 hydrogen bond donors and 1 acceptor, it fits really nicely into this cavity. And we've also subsequently shown that chloride binds really nicely in here as well. And we can show that using NMR spectroscopy. So again, if you're not familiar with NMR, all of these lines on the spectrum essentially mean a proton, so a hydrogen atom somewhere. And what we find is that each of the hydrogen atoms across this face of the molecule are all significantly affected. So as you titrate, as you drop in additional chloride, this one here is the best example. This is the NH here signal. As you drop in chloride, so 0 to 1 to 2, 3 equivalents of it, you see a downfield shift of those signals, which is telling us that the chloride is just binding really tightly within that pocket. So we can quantitatively put that value at around 200 molar. It's not the best, so it's not the tightest binding that we've seen in the literature, but it's up there with some of the best. And actually what we see is it doesn't really matter. So that what you want in effect is something that binds strongly enough to be facilitated through the membrane, but also needs to be weak enough that it will let go of the ion on the other side of that membrane. We looked to colleagues in Sydney, so Phil, to do what we call anion transport assays. So these are vesicle assays whereby we will make a POPC vesicle, we will load it with a high concentration of chloride, and then we will drop it into a solution that contains no chloride and just nitrate. And outside we have a chloride selective electrode, and essentially what you hope to happen is that you get an ion exchange. So the receptor carries a nitrate across, and at the same time it transports a chloride out to I guess, remove the ionic gradient. And essentially once chloride leaks out of the vesicle, you can sense it and you can measure that efflux, as they call it. So it's effluxing the chloride from inside the vesicle out of the vesicle. And essentially this is what you see when you've got a very effective transporter. Over time, you see an increase in chloride in the extracellular solution, and that just shows you that the chloride is being facilitated out of those vesicles upon introduction of your transporter. And again, you don't need to worry about these numbers. What we found was that one of our molecules in particular was a very efficient anion transporter. So this is, again, amongst the most efficient reported in the literature. Even though the design of the compound is significantly different in the binding pocket, we're still getting very efficient anion transport leaking from the vesicle. So we've confirmed at this point we can make it. The structure is what we expect it to be. They're very good at binding chloride, and they're also very efficient at transporting chloride. And as I said, and anyone who was here this morning will have heard Kevin. Kevin's a real expert in not only the antimicrobial and antifungal side of things, but also in the proteomics analysis, which he has been really helpful in doing for us. But Luke, who did this work, went over to Kevin's lab, and they screened against several different types of bacteria, fungi, both gram-negative and gram-positive bacteria, and a couple of fungi. And again, what we've found very quickly was that compound 2, which was the one that was the very efficient chloride transporter, was also a very effective at reducing viable cell count. So while it didn't work in the gram-positive bacteria, it worked extremely well in the gram-negative. So Staph aureus, and in particular methicillin-resistant Staph aureus, were really reduced in viability upon treatment with this molecule. So, and we're getting IC50 values close to clinically relevant values as well. So we're quite excited by this. But again, like I said at the beginning, we sort of wanted to connect the dots between, okay, it's a good anion transporter and it's also a good antimicrobial. What's happening in between? Or can we confirm the fact that it is this chloride transport that's giving rise to the activity? And so the antimicrobial susceptibility assays were repeated with those model compounds, and they found that they weren't active. So these things now are very anti-mammalian cells, so they're toxic to mammalian cells, but not so toxic to antimicrobial cells. True center effect of being involved in the SSPC is I have access to people who have expertise that I wouldn't necessarily have access to, and Finbar in the National Institute of Cellular Biotechnology, who's in Molecules 3, along with Kevin, ran some confocal microscopy with us. And again, the beauty of the compounds are they're fluorescent, so we can track their uptake. You can't see much here apart from green dots, but suffice is to say that the compounds get into the bacterial cells. And what we also noticed was that the cells tend to clump. And so Kevin will tell you that clumping cells, this adhesion, is a protective response that cells undergo when they are under stress. We could also confirm that chloride is being significantly affected here. So we use this MQAE chloride indicator dye. So we treat the cells with MQAE. It's green fluorescent. And when you add the transporter, you see a steady decrease in the MQAE fluorescence. And this decreased level of emission is because you're increasing the concentration of chloride in cells. So we can now, for the first time, say for sure that in the presence of the transporter, the chloride concentration intracellularly has been increased significantly. And that can only be, if you're trying to kill cells, a good thing. This is my favorite experiment. So here is a classic cellular respiration dose response. So the more compound you give it, the more of the cells you kill. If you remove chloride, so if you grow it up in a chloride-free media, absolutely nothing happens to the cells. They survive as if the compound isn't in there at all. And the same thing if you remove sodium. So this seems to be a sodium chloride effect bringing about the toxicity as facilitated by the receptor molecules. And again, just to finish off, well, close to being finished, is again trying to see, with the advantage of Kevin's expertise in proteomics, we could tickle the bacteria slightly. So you give them enough of the compound to stress them, but not enough to kill them. They will then overexpress or underexpress several different proteins, and proteomics can pull out that data. And again, PCA, the principal component analysis, straight away showed that there was a big gap, or big difference between the control population and the treated population, which again tells us very quickly that these transporters in the presence of chloride are having a major effect on the cells. Similarly, again, lots of proteins present. 228 of these were statistically significant and differentially abundant. And there was a really large proteomic shift in response to 2. There's just a few examples. I'm not going to go over the details in the interest of time, but lots of upregulated proteins associated with cell membrane stabilization and organization, self-adhesion as that stress response, cell redox and oxidative stress responses were also upregulated. And things that were downregulated were lipid breakdown at membrane. So these things seem to be trying to stabilize the membrane. It's clear that something is happening at the membrane. And also external virulence factors are downregulated as well. So they're becoming less virulent as they try to protect themselves from whatever is happening to them. And again, the proteome of S. aureus, as just to look at it as a string analysis to see the pathways, what we see is amino acid biosynthesis, virulence factors are all down-regulated, whereas things like transmembrane glucose transport, self-adhesion, and clumping factors are all upregulated as the cells try to figure out what's happening. Another key piece of work that was done again by Finbar was, okay, well, if they're going to kill bacteria, they're probably also going to kill mammalian cells. And interestingly, what we find is, yes, they are toxic at a certain point, but at the IC50, for the bacterial assays, they're completely non-toxic. So at 2.5 micromolar, it kills almost all bacteria. At 2.5 micromolar, it almost does nothing to the mammalian cells. So there's something nuanced in the structure that we're still not entirely sure what it is yet, that makes them much more active against bacteria than against mammalian cells. But that's a very important finding, given we're going to, given you don't want to kill the bacteria, but you don't want to kill the person. And again, anyone who was at this morning's track will have seen Kevin's talk, where he spoke about these Galleria melanella larvae, which show very many similarities with their immune response, or their immune systems as with ours. When we inject them with the compound, there's no toxicity observed. So these tolerate the compound very well, which again goes to show there's something that means that mammalian cells don't get the same effect as the bacterial cells. And similarly, and quite excitingly, when you infect them with a staph infection, so you give them a staph infection, allow that infection to establish, and then treat them with various concentrations of the compound, we see an increase in survival. So again, not everyone is surviving, but we're going from a 26% survival up to a 60% survival over 96 hours. So there's, again, a statistically significant effect upon treatment with this compound. And again, sorry, just after lunch, but this is Luke sent me this picture. It's not statistically relevant, but it's a good visual representation of, they melanize as they die, so they turn black and die. So you can see in the control experiment lots of dead larvae, whereas in the treated population, not so. So not good if you're a moth, but good if you're a PhD student looking for a positive result. And so again, just to show, I suppose, the effect that the SSPC has had on our research program, this is a classic example. So we just last week had the work published in Chem, and it's a culmination of 5 different institutions across 3 countries with authors of 8 nationalities and 4 years of work, and it's led to a really high impact publication. And it's going to come out in the November issue to coincide with World Antimicrobial Awareness Week. So we're going to submit this kind of artist's impression of what's happening to hopefully get the cover. But that's the plan at least. But yeah, it's just like I say, I think this is a classic example of SSPC having a really positive impact and looking at something really very fundamental all the way through to something that's quite applied. And so Luke, you can visit Luke's poster. He is talking about the kind of follow on from this now, where we're looking at various different strands of changing out the structure of these things, adding things to push it more towards bacterial uptake than mammalian uptake, and then looking at adding additional binding units to try and increase that binding affinity. And then that just leaves me to thank the group. So Luke, who's over here, has done a vast majority of the work that I showed you, along with Lokesh, who has since left the group. I'd like to thank all my group, all of my collaborators, all of the people who gave the money, and all of you for your attention. Thank you.